Today we'll continue talking about DNA in which we'll learn about how DNA replicates or makes a copy of itself. So when Watson and Crick came up with their explanation of the structure of DNA, they noticed that the structure also suggested a possible way that DNA could make a copy of itself. And so they suggested that it could be this way, and later experiments proved that they were correct. DNA replication is what we call semi-conservative. Basically what happens in the process is the DNA two strands separate, kind of like unzipping a zipper. And then each strand is used as a mold or a pattern to make a complementary strand, specifically pairing up the bases according to the base pairing rule. And then at the end, when you have two new molecules, each new DNA double helix was composed of one old strand and one new strand. And so we'll see how that works in the following discussion. Here we have the parent molecule of DNA. You notice the structure given there, A pairing with T and C with G. So basically DNA would unzip. There would be free nucleotides that would attach themselves into place step by step. Each strand is serving as a template, and then you end up with two identical daughter molecules. Notice that each one is half old, the blue, and half new in the gray. Of course, it doesn't happen quite that simply. There are enzymes involved in the process, and so we'll discuss how that works. Here we see kind of the process as it occurring in the ribbon model. So the DNA unzips, and then the nucleotides are matched up, producing the daughter DNA molecules. DNA replication occurs in what's called the 5' to 3' direction. That has to do with which sugar is on the top, or which carbon is on the top or the bottom of the sugar molecule. You may or may not have noticed in previous models that the sugar molecules are facing up in one direction and down in the other. And so since the sugar molecules are in opposite orientation, then it has to be done just a little bit differently on one side and then the other. So there are two important proteins that are involved in this replication process, and these are DNA ligase and DNA polymerase. DNA ligase is kind of like tape in that it joins the small fragments or small pieces into a continuous chain, whereas DNA polymerase is the molecule that basically adds the nucleotides to the chain and then proofreads and corrects if bases pair up the wrong way. The DNA polymerase and DNA ligase also repair DNA that is damaged in some way by things like radiation and chemicals of various kinds. The DNA replication makes sure that all of your body cells in a multicellular organism have the same genetic information, because we want every cell to have everything it needs to be part of the same organism. Here we have something that shows that this is occurring in many places at once. Here's our parent DNA molecule to begin with. There's several origins of replication where the DNA unzips, forming a bubble, so to speak, and the DNA is copied within each one continuously until the bubbles connect with each other and enlarge, and then you end up at the end with two separate DNA molecules. This shows the diagram that shows the 3' and 5' orientation of the molecule. Here you can see the sugars. The carbons are labeled 1, 2, 3, 4, 5 there. And so this is the 5' end, and this is the 3' end of this side, and then the other side is oriented the opposite direction. The sugars are upside down from each other. And so it copies from 5 to 3 on this side, but the other side has to copy in the other direction. So it makes it a little bit more complicated when you have to do them both at the same time. This shows how this is going to work. On the 5' strand, that can be copied directly from 3 to 5. But on the other side, it has to do piece at a time. And so basically, the DNA polymerase pulls out a piece that copies in the backwards direction, and then pulls out another piece that copies in the backwards direction, and they get attached to each other. And then ligase comes behind and attaches the fragments together. And we'll see a video in class that'll show how this works and make it a little bit more clear.